Hello, everyone. Sorry for the late start there. I just wanted to wrap up some final things, but welcome to today's webinar. My name is Mike O'Brien, and on behalf of Trelleborg Healthcare and Medical, I'd like to welcome you to today's webinar on introducing active pharmaceuticals to silicone and controlling elution rates, presented by Zach Fletcher. Zach is our business development manager for drug combination devices, and he is located in Minneapolis, Minnesota, and he works closely with our Tustin, California facility, which is where we handle all active pharmaceuticals. We also have Steve Hong with us today, and Steve is our principal engineer located in Tustin, and he'll be joining the Q&A session later today. Nice to see both of you guys.
Hi, Mike. Good to see you.
So, very brief introduction from me. So I think it'd be a good time here for the both of you to just tell the audience a bit more about yourselves. And Zach, I figured we'd start with you on this.
Yeah, no, it sounds great. Yeah, so I've been a part of the Trelleborg team for a little over a year here, but I've spent the last 8 years in the manufacturing space. So previously with a company called Trelleborg, who many will know, previously with Protolab, currently with Trelleborg, and yeah, been a part of the team for a little over a year and enjoying the drug-eluting space.
Thank you. And Steve, do you want to give a quick intro?
Sure. Yeah. Hi, my name is Steve Hong. I'm the principal engineer at Tustin site. I do have about 15 years of pharma background. Right now, I've been with the Trelleborg for about 3 years, focusing on the combination products.
Great. Well, thank you both for that. And to our audience, before we get started, I just want to point out a few things just to make sure you know how to participate in the webinar today. So first off, we highly recommend questions for our speakers. You can submit these through the questions pane of the control panel, and feel free to ask them at any time during the presentation. I'll collect them and we'll address them at the end during the Q&A session. And please note that we can't see or hear you, so if you need to communicate with us, please use the chat window and we'll get back to you and help you out however we can. And one final thing is that this session is being recorded, and we'll send you a link to the recording once that's available. So with that, Zach, I'll pass it to you and let you take over.
Sounds great. Thanks, Mike. And I'll turn off my camera here so that we can focus in on the content. But I'll start off by just saying thank you to everyone in attendance, those who are joining live, and those of you as well from the future who may be viewing this recorded session as well. So just a quick welcome and thank you for your time. So the main focus of today's conversation really will focus in on the work that Trelleborg does in drug elution for combination products as well as implantable drug delivery systems. We've done a lot of work in this space for quite some time, and looking forward to sharing a little bit more info with everyone here today. So to start, I'll quickly note that we are actually basing much of the session today on a white paper that we released back in April. So hopefully some of you in attendance are familiar with it, have read through, and might have even brought some questions for us to answer as follow-ups to the content presented within. And then I'll also note that if you haven't read the white paper yet, you find yourself leaving today's session looking for some more detail, we'd also highly recommend that you download and review it. Mike, I'll shoot over a link to you here that we can drop into the chat for all the attendees, just so that they can get at the information. So I highly encourage anyone to follow that link and be able to download the white paper to get a hold of some of that content. So just looking through today's agenda, we'll start with a quick introduction to drug elution. What are the advantages of the technology? We'll cover some of how it's being used in the market today. We'll talk through some of the market expectations as well there, and then we'll jump into more of the manufacturing considerations. So we'll talk through some of the excipients that are used, our manufacturing methods, and how we'll actually formulate and combine the pharmaceuticals with some of the excipient materials into these systems. And then lastly, we'll spend a little bit of time going through some of the case studies that Trelleborg has executed in the past. So we've done a significant amount of work in the space to continue to inform our teams and generate learnings that we hope the space will eventually use. So we'll spend a little bit more time on each of those and hopefully generate some learnings for those of you that may be in the space already today. So with that, let's jump into just a quick overview of drug-elution, how it's being used. We always kind of start that conversation. I always like to start that with the impact to the patient. So, what kind of advantages can we generate for the patient, and what kind of impact can we have on their quality of life? So to do that, to try and frame some of that up, I've put together some patient statements that really could be made if we were to continue to push forward these technologies. So I'll quickly walk through those so all of you can get a feel for that. So first one is, I no longer need to remember to take a pill every day. I'm able to make fewer visits to see my care providers. The side effects I've felt have been greatly minimized. I have more confidence because I know the procedure that I'll be going into is routine. My body seemed to recover faster following my procedure, and I spend less time managing my condition. So all of these really boil down into some of the key themes that are mentioned here in this slide. Patients have the ability to spend less time managing their condition. There's a greater potential for them to minimize the side effects felt from the therapy. And then there's also just a greater potential to generate and provide minimally invasive therapies to patients as well. So quite a big impact that can be felt by the patient themselves. Now, when we look at it from the pharma side, or maybe more of the industry side of things, we look closely at three trends. And these are certainly not an exhaustive list, but from my standpoint, I feel pretty highly impactful trends that these kinds of therapies can actually address. So the first is targeted delivery. This is a theme that's emerged quite significantly in recent years. The ability to put the therapy or a drug in close proximity within the body to where that therapy is needed can be highly impactful. Through that, you're able to really drive towards lower dosages of those drugs into the body because of the ability to really target it. And through that, because it's localized as well as a lower dosage, you can really drive out a lot of the potential side effects. So targeted delivery is a big one for us there. As well, with the ability to really drop down the dosage, it also increases our ability to extend the release and formulate something that drives towards a more extended release of a drug or any kind of therapy with a single dosage. So it becomes possible for us to extend delivery of a drug into the month or many year range. So it becomes very possible for us to provide that single dosage that then allows patients to not have to continually manage or take any kind of drug within that process. And then lastly, patient compliance is always a significant challenge from the standpoint of what we can actually do with these therapies. Again, because you're able to extend that release, you're able to overcome challenges as far as high risks of non-compliance. Maybe there's advanced age that makes remembering to take a pill challenging, or there could be situations where psychological state may drive towards less of a likelihood of patients taking a pill every day or on a regular basis. And so we're able to combat that quite significantly with these kinds of systems. And then there's also the advantage that really can be driven forward by providing a therapy that combats maybe a high level of maintenance condition. You think about something like diabetes, where you're constantly having to think about it, manage that condition. If we're able to generate a therapy that is a implantable single-use system that allows for a lessening of the maintenance that goes into managing that condition. So all of these really drive towards obviously a great ability to impact the lives of patients dealing with some of these conditions. So if we look at what the space is doing today, there's a couple different use cases, or at least how I bucket these. The first case in kind of the early stages of the use of these types of systems, I call medical device biological acceptance. So this is where the device itself is actually the primary mode of action. So you have examples of things like drug-eluting stents, drug-coated balloons, pacemaking leads with a steroidal collar. All of those are really able to be supplemented by the introduction of a drug, be it an anti-inflammatory, antimicrobial, antibacterial, even antifungal type applications. The efficacy of those devices can be driven up and improved by introducing some of these drugs, and that trend will continue going forward. But the real potential, I think, and what we'll start to see emerge in the future is implantable drug delivery systems. So you see some examples here of some well-established systems in contraceptives that really served as some of the early examples of this. But I think going forward, we're also going to see more conditions be addressed by some of these systems, and there's great potential to directly address some of those conditions with these types of systems. So a big area of focus, and part of what makes Trelleborg and myself so excited about the future of these technologies. And we're not alone in that. So looking at what the market actually expects, back in 2020, it was estimated that revenue from implantable drug delivery systems was approaching about 19 billion. Within 5 years, so we are now 3 years from this projection, we expect that figure to be pushing a $30 billion mark. And so when we start looking at the growth expectations of something like this being in the 10% range, again, that's a pretty massive growth expectation. And as we go forward, the challenge for manufacturers, and really why Trelleborg is putting out this content, is to be able to continue to evolve the manufacturing processes to support the development of many different pharmaceuticals into these kinds of systems. That will be the manufacturer's challenge in being able to help support and achieve some of these market expectations. So we're excited for that, and it's the main basis for why we are covering some of the content we share here today. So with that, we'll jump into our first poll question. Back to you, Mike.
All right, thanks, Zach. So we have a few poll questions throughout the presentation today, and you go ahead and respond by just simply clicking on the screen to select the option that best represents your view or your opinion on the question. So this first one is asking, what is your experience with drug-eluting products? And we're asking you to select one of these. Either you're actively involved in developing drug-eluting products, you're familiar with these therapies, but they're not a focus, you're not aware, but you want to learn more, or you're just here because it sounded like an interesting topic. So we will give here a few more seconds. It looks like we've got the majority of folks have responded, but we'll give you another 5-10 seconds here to see what comes through. And we'll share the results. And again, these are completely anonymous. These display as percentages so we can get an idea of the audience breakdown. All right, we'll close that and share here. So it looks like we have a decent break here. We have 39% who are not aware but want to learn more, 28% who are actively involved, 22% just like the idea of the topic and want to learn more, and then 11% that are familiar with the therapies, but are not one of their focuses. So thank you very much for the participation there. We'll have 2 more polls coming up, and I'll share once we get there. And Zach, you can take it over again.
Awesome. Thanks, Mike. All right, so now we'll jump into more of the manufacturing considerations. So we kind of think through a lot of these things really as the options that developers of these systems might be able to consider. And I think, you know, my main point here is that the great thing is that we do have options, right? And so we'll talk through some of the considerations there and really some of the processes that Trelleborg uses to generate some of these systems. So one of the first considerations really are excipient materials. What are we putting the drug on, or what are we mixing it with? And to date, there are essentially 2 dominant excipient material classes in use today. Those are silicone and bioresorbable polymers. Each of these really has its own unique kind of place within the system and or within the space, and you know, we'll kind of talk through what some of those are. I'll say that Trelleborg's main area of expertise really has been in the silicone side of things, but as we go forward, as we continue to add to our team, there's a recent acquisition that Trelleborg made of a company called Immerse that we will also be bringing in some of expertise in the bioresorbable polymer side of things as well. So quickly working towards being able to serve you know, our customer partners with many different options as we move forward. So but just to highlight considerations here within this space, first and foremost, silicone, it really is more of the dominant material, excipient material in the space today, and that really comes down to just the ease of being able to manipulate the chemistry of these materials, and ease in being able to actually manufacture in these materials. So there's a great potential with these materials to really extend the release. Everything that I've seen does suggest that silicone materials really are able to push forward more of an extended release system or window for these therapies. And that is a bit of an advantage here for us in those materials as well. Some of the potential downsides I'll note here are that it doesn't break down in the body. So there certainly may be situations where you know, the used drug core can actually remain in the body following the exhaustion of the pharmaceutical itself. But there may be situations where that implant may have to be removed at some point. So just a consideration when looking at some of these systems. The opposite is true, and it is somewhat the advantage for the bioresorbable polymers. So these materials will break down in the body, and at the same time, even though they break down, they would still be eligible for early removal if there's a dosage challenge or something like that that needs to be dialed in for the patient. So there are certainly some advantages there. Some of the downsides with these materials or considerations maybe is that it's a little bit more difficult to chemically modify, and there are more challenges from a manufacturability standpoint. So you know, depending on what the system requires, will typically drive us towards what's the best approach there. As far as manufacturing methods, I'm excited to note that, you know, really the dominant manufacturing processes that exist in the space today can still be leveraged. So you do still have the ability to mold, extrude, and sheet into different you know, components, and all of those can carry the drug element. So there really aren't too many limitations from a manufacturing process standpoint or a manufacturing you know, standpoint. So all of that should be available. I list 3D printing here as well. There certainly aren't any active or approved in market systems that are leveraging 3D printing, but obviously this is a trend that exists everywhere in manufacturing at this phase, and there's been quite a bit of research actually put into how we could actually formulate some of these therapies in a 3D printing process. So I do expect that to emerge in the space as we go forward as well. And then I list other two just because the three listed above really are where Trelleborg is playing in the space today. But we don't see this as a completely exhaustive list. Hypothetically, you should still be able to manufacture in other ways as well. These are the ones that we're most familiar with here. So then the final consideration for how we're formulating a lot of these therapies gets into how do we combine the pharmaceutical component with the excipient component. And Trelleborg uses 3 different systems or methods today. I'll highlight some of these icons that are shown here at the bottom as we make our way through the rest of the presentation. You'll see these icons typically top right of the slides as a reminder of which process or method we're referring to within that. So the 3 are mix and manufacture. There's the immerse and impregnate approach. And then we also have the ability to enclose within a membrane system as well. So as we go forward, I'll do a bit more of a deep dive on each of those systems. So we start with mix and manufacture because this is generally the best accepted method. It's the one that's in use today. And it really comes down to mixing the excipient and the raw material together prior to the manufacturing phase. So I'll provide a little bit of an icon process here down at the bottom to kind of show how that process works. Basically, you're combining the pharmaceutical and the excipient materials together prior to the manufacturing phase. You'll mix those and then push it through the manufacturing process. And at the end of that, obviously, you've got your finished product with both drug and excipient material formed. There are some downsides to consider, or maybe some drawbacks in situations where the pharmaceutical or the drug compound may not stand up to this process. And that was really come down to heat sensitivities of the API compound. So it may break down at the manufacturing temperatures required to complete that process. And there also may be some obstruction of the curing process with the drug compound being involved there as well. So a couple different scenarios where that may not be possible. And that really is what drives us towards considering the alternative methods that we talk about here the rest of the way. So as a viable alternative where we run into some of the challenges previously mentioned, we do have the ability to immerse and impregnate a drug within the system. So again, following the visuals provided here, this is where we're taking the excipient material and actually pushing it through the manufacturing process without the pharmaceutical involved. Then we go into a process where that finished product is then combined with a solution. So you're combining the pharmaceutical ingredient with a solvent and creating a solution. And then once that is done, you immerse the silicone sample or potentially another excipient material into that solution. There's a swelling that occurs with that sample, and then that allows the pharmaceutical to basically we'll refer to as impregnate that unit. So once you remove that solvent from the system, you're left with a finished component that is both device and drug. So again, the advantages of this is that we're introducing the heat for the manufacturing process, and we're completing the curing process prior to the introduction of the pharmaceutical. And that's really where there is some place for this process within formulation here. There are some other considerations here. This process will tend to favor a bit more of a burst release profile. And there's also greater potential for variance within the concentration of the drug. So again, considerations as we look at if we want to commit to a system like this or explore it further, but there certainly are situations where it does make sense for us. So then the last method is enclosing a drug core within a membrane. So again, using some of the visuals here, that we have below, I kind of show either or, because this can be used as a system or a method for controlling the release rate, where we have a solid drug core, either in a solid core, maybe it's crystalline type structure, or there have also been situations where you have the drug in a pill form, and you're putting a membrane around that bit of material. But I also highlight and use the same icon from the previous slides as well, because this method can really be used as a modifier to better control some of the results that we're getting from the previous two manufacturing processes as well. So we're able to apply a membrane and really use that to better control some of the results that we're getting through this process as well. So essentially, you're introducing a new component within the assembly, and that does allow for the control of the release of that profile. Obviously, introduction of a new component does require maybe a little bit more manual effort to apply that material within the assembly. And with that, there's considerations from a cost and efficiency standpoint that need to be considered as well. But once again, if that is something that's needed to control and drive towards the correct elution profile, then that is something that we can do as well. All right, and with that, we'll jump into our next poll question.
All right, thank you, Zach. Great stuff there. All right, our next question, we are asking, which of the following challenges seem most addressable or impactful via implantable drug delivery therapeutics? The options are providing targeted delivery, extension of drug release, addressing patient compliance challenges, or other. And if you select other, you can share comments or thoughts in the chat feature if you'd like. And again, you go ahead and click on the screen to select your answer, and I'll give it another 10, 20 seconds here to let some more responses come through. Right, we're still getting a few more here, so thank you for responding, and I will go ahead and share these here shortly. 1 second, please. All right, so looking at these, Zach, it looks like the targeted delivery and the extension of the drug release are the two challenges that seem most addressable, and then we did have a few for addressing patient compliance challenges as well. So again, thank you for that, and back to you, Zach.
Awesome. I appreciate all the feedback on that as well, everyone. All right, so now for the rest of the way, we'll focus in on some of the case studies that we have generated through some of our own effort here. And so since many of the established methods really are that mix and manufacture approach, that's kind of the best established method and the one that Trelleborg is most familiar with. We've really committed to exploring some of the alternative methods. So what you see on the screen here are the 3 case studies that we'll make our way through the rest of the presentation here today, along with those icons that I mentioned as well. So the first 2 case studies will deal with the immerse and impregnate approach, and then the last one will round out, focuses more in on the membrane approach there as well. So just as a quick overview there, we'll look at first the immerse and impregnate approach really as a process technology platform. So we'll spend a little bit of time with that, making sure that you're able to understand how that process looks and what kind of considerations go in there. The second one will focus in on, do we have the ability to actually impregnate multiple pharmaceutical ingredients within this process? So really, it's a focus in on the ability to introduce complexity into some of these systems. And then we'll round everything out with a deeper dive on the introduction of membranes and how those can be used to really control the elution profiles for some of these systems as well. So looking at the first one, as you know, immersing and impregnating sort of as a platform technology. Our team sought out to achieve essentially the highest possible concentration for 4 different drugs through this process. So as we make our way through this case study, we'll share some of the important considerations in designing some of these systems, and really how all of that feeds into designing an effective system as far as introducing these kinds of components and materials into the systems. So at the end of the process, we did use a gravimetric analysis to evaluate the samples that we produced and really test out what the results were. So it's essentially a comparison of the sample weights pre and post immersion process. And our team took 35 durometer silicone samples. Started out as 20 millimeters in length, and essentially were placed in vials of each of the solutions for variable lengths of time. So as you can see here, that vary between 5 minutes and 6 hours for immersion time. So as we jump into the next phase, before we get to considering the pharmaceuticals that we're putting into these systems, we need to look closely at what each of the solvents that we could potentially use do to the samples. So we mentioned previously that the silicone materials really do swell within each of these solvents, and to varying degrees. So it's important to understand that and how that's performing in each of these systems, so that we can effectively design a process for doing that. So here's a quick overview of what we saw in some of that testing. It was interesting that we had 2 solvents that actually increased the length of these silicone samples by about 50%. So 2 that really stood out as an ability to increase the size and swell the material. There were 2 others that ended up at about a 15% range, while all of the others did increase the length of the samples by 10% or less. And as a control, we also did evaluate the effect that water had on the silicone samples, which was 0, as it should be. So just using that to confirm that there wasn't anything else kind of going on within that, and really prove out that we have good results from this. So then our team sought out to really understand what's the maximum concentration of each of these compounds that we can fit into the solvents. So the solubility of each of the drugs within each of the solvents. So what our team did was, at a consistent rate, the pharmaceutical ingredients were introduced to the solvents in powder form, and continued to be mixed until we could actually see that solubility, the maximum solubility, was really being challenged. And you can confirm that by just seeing some of the undissolved powder within the solution. At that point, once we did see undissolved powder, our team would add just a little bit more solvent into the mixture until we returned to a clear solution state. And that was essentially how we reached these maximum solubility figures. So for each of these pharmaceutical plus solvent combinations, our team then took the greatest concentration and advanced those forward. So for each drug, we took 2 of those into the next phase and completed the information that follows here. So let's take a look at the results from this effort. And I think the major item to point out here was that our team was able to successfully introduce a measurable dosage of each of the pharmaceutical ingredients in 6 out of the 8, from 6 out of the 8 solutions. So ended up at about 75% success rate. And I'll also note that all 4 of the pharmaceutical ingredients, we were able to introduce some kind of measurable dosage into the systems. And those concentrations for the successful ones did range between about 1% and 6%. So if we look closely at the table and really what it says, the first column here deals with just listing out the solutions, and then once again reminding what kind of concentration of the pharmaceuticals into the solvents we achieved And then we list out 3 different weight measurements. So the first is the initial mass of the silicone sample prior to the effort. There was another measurement taken 2 hours after the immersion process was completed. And then there was a final mass taken 24 hours after the devolatilization process was completed. So an interesting learning here was that the maximum swelling and maximum drug load combination did not necessarily equal the highest resulting drug load. So we were successful with the combination that swelled the silicone samples by 50% and had the greatest concentration of drug and solvent mixture, but it ended up being the concentration that was the lowest of the 6 that we were successful with. So an interesting learning there that it's not all about how much can you swell the material and how much drug can you get into the mixture. And then a second interesting learning was that the increase in the mass of the sample, so at the measurement we took at this 2-hour after completion phase, that didn't exactly equal the new mass introduced based on the percentage of the drug that was in the solution. So what we would have expected is, for example, this first one, the increase in mass that was experienced should mean that at the end of this process, we should have gotten, about 31% of that drug into the system based on the mass increase. And what actually happened was that the amount was something less than that. In some of our observations, that ended up being about 54% of the anticipated drug did not actually, or was actually captured within the system at the end of that process. So our team came to refer to this as a bit of the partitioning effect, which is something that will have to be accounted for as you look at some of these systems. And it also does help to have a really close relationship with the material provider, the excipient material provider, to try and modify some of the chemistry of those materials to really drive towards positive results for something in this process. All right, so jumping into the second case study, again, this was the one where we're looking closely at, can we introduce multiple ingredients, multiple pharmaceuticals into an existing system, and do you have the ability to control how much you're introducing, and is it truly possible to introduce multiple? So there were 2 antibiotics that we selected and attempted to impregnate into a single extruded silicone sample. And we used basically hydrocephalus shunts as an example of a product that was in the market. We call this a predicate device to really generated the targets that you see bottom right hand side of the screen here. It gave us a target to pursue as well as an acceptable range of the drug concentration within the system as well. So we took both of the pharmaceuticals and mixed them with chloroform as the solvent, and then immersed silicone samples in that material, that solution, for anywhere between 5 and 120 minutes. And in this situation, we assessed the results with HPLC equipment that helped us understand how much drug we have in those systems. So if we take a look at the results, I'll quickly describe what we're seeing on the right here. So each of these lines with the points does represent a different solution that was tested. So those are important considerations. They're basically the results that we received. And both of these are the X and Y axis is the concentration of each of the drugs within those systems. And then also the main target that we were pursuing based on the previous slide, the targets that we were looking for, as well as the ranges are represented here by this point with the white X in the middle. That is the true target that we were pursuing. And then this green box actually is the range window, essentially, that we're targeting. So once we have all of that kind of set up, our early attempts here really were some of these black lines and more of the gray lines. And we had to improve upon this, obviously, because in the first situation, we weren't quite able to achieve the drug concentrations that we wanted to or needed to for those components. And then there's also the challenge in the second scenario where maybe we could get close to the window, but it wasn't quite well centered. And when you see a huge range of outcomes here, that can be difficult to control as well. So our team, after those initial attempts, did go back and make another attempt at improving upon those results. And that's where you see now these two systems that were also possible that our team generated as results. So the red circling here really is a highlight that we were able to achieve, and these are two different solutions. We were able to achieve results that were very close to the target. And I think that's the main thing to point out here, is that now when we have two different solutions that are capable of generating something closer to the range, then you can quickly jump into looking at each of those maybe more in depth and talking about what the target of the project ultimately is, so that we can focus in on where do we go, where are we investing, what kinds of questions are we asking to continue to develop this process and make sure that we're designing an effective system going forward. And then the final piece to note here is that we did complete some testing following the production of these samples to really prove that there wasn't anything that really challenged or inhibited the effectiveness of the antimicrobials after this. So the introduction of the solvent or the application of them within silicone didn't change anything about how they performed. They still did what they were supposed to do at the end of that process. All right, and then lastly, the third case study focused in on long-term effectiveness of silicone membranes and how they can be used and employed to control the elution profile of a system. And so our team really wanted to make sure we understood what factors, what levers can we pull when we're looking at these systems, what factors are critical in the design of the membrane, and really wanted to get towards how do we effectively design for these kinds of products. So to do this, we actually took a look at a predicate device once again, and with samples of that device, a 28-day release profile was taken, and that essentially became the target that our team pursued. And so we proceeded into manufacturing our own samples and then making changes to the concentration of the API within those systems. And then when it comes to the reservoir, looking at the durometer, which would refer to the type and size of the silicone reservoir that's going around the drug-loaded core, as well as the wall thickness of that membrane, and then the degree to which the cross-linking has been completed as well. So all of those were factors that were considered as we designed these going forward. So if we look at what we found in the results, first I'll quickly describe this graph that you see on the right. The predicate device with this gold line was essentially the target that our team was pursuing. So that was basically what we were seeing for the device that was on the market. In our first attempt, we ended up coming up with a system that performed close to the predicate device. It was within the right window, but as you can see, it ended up performing and releasing a little bit more of the drug than the predicate device did. So after that initial attempt, once again, just like in the previous case study, we did take another stab at putting together a system and designing a membrane that would more effectively control that elution rate, and came up with something that was, as you can see in the blue line here, much more closely aligned to the concentration of drug that was released over time, and actually proved to be more consistent than the predicate device as well. So obviously through that, there were some major learnings that were driven out of this. One was that the drug concentration certainly had an effect on the release rate, but it was somewhat minor when compared to some of the other variables. So our team actually found that the thickness and the durometer of the membrane really had the largest impact on the results. And the wall thickness was actually quite sensitive. So all the way down to about 5/10,000ths of an inch variance within the wall thickness would drive detectable variation in the release rate. So for us, this really drove home how impactful precision in these systems is. If there is the ability to have some variation by about 5/10,000ths of an inch within a membrane, that can actually drive variance in the release rates that you'll see. So it becomes crucial to be able to control down to that level for some of these systems. But you know, as you can see here, as we continue to work on and design for some of the systems that you know that we have here in pursuit of that predicate device, our team was successful in driving towards a system that was even more effective from the predicate device, from the standpoint of consistency in a release rate of that drug into the body or into a system. All right, and with that, we are to our last poll question.
All right, thank you, Zach. Like you said, our final one here, and we'll try to get through this quickly so we can get to questions. But just pretty simple one. What was the most interesting topic covered? Is it the market potential for these drug delivery forms, the applications where these technologies are being used, the detail provided on the alternative manufacturing methods, or if other, again, you can feel free to share comments in the chat section. So we'll just give this one a few seconds here so we can get some info and get to those questions for you. Right, we'll close this out and share very quickly. So again, we have a good mix here, and it looks like the majority selected the detail provided on the alternative manufacturing methods. At 29%, we had the market potential for these drug delivery forms, and at 12% apiece were the applications these technologies are being used and other. And I can go ahead and monitor the comments to see if anyone put the reason for other in there after the fact, but that closes our polls for the question, or for the webinar, sorry. And Zach, I can go ahead and pass to you.
Yeah, sounds great. So just wanted to close out and say, obviously we covered quite a bit here today. Thank everyone for their time and attention. And you know, if there are other questions or you want to learn more about other things we're doing, I'll share the website here where you can get to a lot of that info. But at this time, we'll look forward to taking any questions from the audience.
All right, thanks, Zach. Let me get to the questions pane here and see what we have. One that came in early on, you may have covered it during the presentation, but someone was asking if you could give more examples of bioresorbable materials.
Yeah, so there's a couple different classes. I'll see them referred to as PLA, PLGA. I think there's a PGA. Those are just different materials that, again, the main focus in what they're doing is just the ability to break down within the body. So as far as I understand, they'll break down to carbons and you know, other basic materials over time. And as that does break down, it's really able to release to the drug into the system as the drug kind of is exposed to the body that surrounds it.
Okay, great, thank you. Another one we have is asking, does Trelleborg have their own HPLC, or is it tested by a third party?
Yeah, Great question. So we do have some of those in-house capabilities as far as HPLC equipment and are able to attest, plenty of the samples and evaluate what we're seeing in some of those systems. We also, in some situations, will outsource based on the test that's required and those sorts of things as well. So at this time, we do have in-house capabilities, and there's also scenarios where we leverage outside resources to execute on some of the tests as well.
Okay, great. Thank you again. We have another one here asking, they're curious to know if there are any market products that use the soaking impregnation method.
As far as I understand, I don't believe there are any yet today, but it certainly is something that as we continue to pursue the market in achieving some of the market growth expectations, I would anticipate that we're going to see some of those emerge over time. And so we think, as Trelleborg, obviously think that it's an important method to continue to consider as we build out some of these systems and offer to device developers or developers some of these systems going forward.
Okay, great. Good question, and thanks for the response there. So we have a few kind of similarly themed ones here, just showing interest in the silicone membrane. So I guess, do you mind explaining just kind of how you control the elution rate with that a little bit and kind of focusing on that aspect of it?
Yeah, and this is where we introduced Steve earlier. He's more, on each of the projects and really can probably shed quite a bit more light than I can on this. Steve, would you mind jumping in and kind of explaining some of the considerations there?
Sure, yeah. So there are two aspects of controlling the elution rate. First thing is the drug core. When you fabricate the drug cores using the silicone and API mixtures, you need to look at the chemistry, and meaning depending on the catalyst type you use, it can actually make the reaction progress to a certain level, and then you need to look into the matrix polymer, the matrix formed within the drug cores. That will actually affect some of the elution of the APIs from the drug cores. But more importantly, it's actually the wall thickness of the release membrane that's being applied to the drug cores. So that's a more critical item to look into in terms of controlling the elution rate and the material that we use to form the release membrane.
Okay, great. Thank you, Steve. All right, looks like we have a few minutes, so we can try to get to a few more here. This one is asking, in what specific applications are you seeing activity, and where do you anticipate others to emerge?
Yeah, great question. Yeah, and I think it's still a young space, so I do anticipate that we're going to see, a lot of evolution in the coming years here. But the main things we've seen so far really have focused more in on, contraceptive systems. Obviously, that was mentioned earlier. That's been a heavy area of focus for many in this space today. And then also, we're seeing quite a bit of momentum in different ophthalmic applications. There's quite a bit of momentum in, different oncology, cancer treatment type therapies as well. There's allergies, ear, nose, throat type applications. And I expect, we're going to continue to see a lot of thought put in these materials as well as we kind of go forward in developing these systems for many different applications. So I do anticipate that this will change quite a bit over time.
Okay, great. And hey, Steve, I'm coming back to you for this one here. We had a follow-up on the membrane question. So they're saying, just to make sure they're understanding, the concept is the membrane dissolves slowly, reducing thickness, and the elution rate increases. And is geometry play a factor on this? So like in terms of like total surface area per volume ratio?
Okay, let's get back to the first question. Does the membrane dissolve? No, it does not actually. The membrane stays and it just remains as it was formed. The second question is the surface area. Yes, surface area is also very important in terms of controlling the elution rate. So the way the shape of the drug course and then the shape that release membrane attached to or applied to the drug core, it matters because that can extend the surface area exposed to the solvent. So that's also a factor to consider for elution rate.
And I can add to that too that there are some more complicated membranes that I've been exposed to or heard about as well, where there's part of the membrane approach is that you can create little windows. So it does essentially serve as a mechanical limiter of the pharmaceutical from the systems. You can apply windows to the membrane. So that is another element that can allow us to somewhat control the elution rate. And maybe the most advanced that I've seen is that some of those windows are sometimes something that does dissolve. So you've got a mix of windows that open up over time that can create a little bit more complexity to those systems as well. So it's not simply membranes. There's a lot that can be done with those systems in pursuit of trying to control some of the elution rate over time.
All right, looks like we're about there. Let's sneak in one more here and then we can close things out. So this one is asking, how do you select the formulation method as being appropriate for the desired application?
Yeah, I think this really does come down to a conversation with our customers on what they need, kind of what stage they're at in development, what drug compounds are they using, what sensitivities do we know about the drug compound, and then also becomes a conversation about what do we see in the future as well, what are the anticipated volumes, what kind of critical factors are we pursuing and designing some of these systems. So I think through all of that, we really have a good, robust discussion about what do we see for the system when we get to final state. And through that discussion, we can start to work towards what is the right approach for us. How do we want to pursue formulating this system, and what kinds of things do we still need to learn about that drives us towards designing experiments that can help us really trend towards developing a robust system that can be used at scale.
Great, thank you. All right, I lied. I'm sneaking in one more here. That came in while there, Zach. I don't know if this is for you or Steve, but the silicone membrane, so is that made of a hydrophilic silicone that water can penetrate?
Steve, I'll defer to you on that one.
Oh, all right, sorry. Okay. The silicone membranes, so does it penetrate? Does water penetrate into the drug course? See. It depends on the API materials. You might not introduce your the drug course or the implant to the water material. It depends on the solubility of the drug APIs being introduced. So you need to choose your solvent carefully. Hydrophilic, is it hydrophilic? It really depends. So that also depends on your the type of APIs that you use, and also the solvent you choose. So it really depends on your application.
And I think there could be situations where both may apply as well. There may be some systems where it'd be advantageous for it to be hydrophilic, and others where hydrophobic is preferred. And so I think in both of those scenarios, it's something that we can explore based on the need of the system.
It's great. Thank you both for that. All right, that is it then. So we've reached the end of the live session and the Q&A session here. So thank you again, Zach, Steve, for the great insights and for sharing those with us. And to our audience, we hope that you found the information that was presented by our speakers today valuable, interesting, and you see that the team in Tustin, California is able to help assist with your application in the near future. The recording of our session, as I mentioned, will be available shortly, and you will receive a link via email as soon as we have that ready. So again, thank you to the presenters, to our audience, and I hope you all have a nice rest of your day. Thanks.
Thanks, everyone.